أهلا بكم جميعا ومرحبا فردا المناعة في زمن الكورونا. معنا ضيف رائع اليوم الدكتور علي برانسي من زيورخ من شركة روش العالمية لصناعة الأدوية. رفيقتي في هذا اللقاء دكتورة أهداب صعيدي. كيف حالك دكتور؟
الحمد لله بخير.
طيب دكتور، تعرفين عن نفسك لو سمحتي؟
بالعربي ولا بالإنجليزي؟
بالعربي لو سمحتي.
طيب، اسمي أهداب صعيدي، أستاذ مساعد في جامعة الملك عبدالعزيز، تخصص علم المناعة، والتخصص الدقيق علم السرطانات والعلاج المناعي.
نرحب بالضيف. Ali, how are you?
Very good, thank you. And you?
Not bad, my friend, not bad. Thank you for joining us today.
Thank you for the invite.
Anytime, my friend. So tell us a little bit about you, Ali. What do you do?
So I'm a scientist at Roche, I think, as you already mentioned. I've been doing cancer immunotherapy for now, let's say the past 10 years. At the moment, I'm enjoying my quarantine at home, still working for the past five weeks. I'm based in Zurich in Switzerland, originally from Canada. As you might have recognized the picture behind me, so this is Quebec City.
So what are we looking at? What's that castle-like building behind you?
So the castle is called the Chateau Frontenac, so the Frontenac Castle. Now it has been converted into a hotel. So actually, if you visit Quebec City, you might be able to take a room there. It's a beautiful place, very European-like for being part of America. Yeah.
Mm-hmm. And so you were born and raised in Quebec.
Exactly, born and raised in Quebec. Then I traveled for studying quite a bit. I have a few slides on this, so maybe should I share my screen right now?
All right, my friend. The screen and the mic are yours.
Perfect. Thank you. So first of all, well, thanks a lot for joining us today, and thanks a lot for the invitation. That's a pleasure to be able to share a little bit of my path and what we know and where are we on therapies for cancer. So I'd like to start a little bit with myself. This one is a little bit in contrast to the background that I have currently on Zoom. This is still Quebec City in the same castle. So Quebec is widely renowned for its tough winters and for maple syrup. So that's part of the things that I'm missing nowadays. But in Zurich, it's still also very enjoyable despite the winters being much easier as well. So I'm glad to be able to evade a little bit the freezing cold temperatures that sometimes dip to -30 in Quebec City, -30 Celsius. So that might shock some people. So I'm very glad to be in Zurich and also to be able to evade it. I have parts of my family that comes from North Africa. So sometimes I get a chance to travel to Tunisia as well, which is a beautiful place. I've been there a few times only, didn't spend maybe sufficient time there to be able to learn any Arabic, unfortunately. But I really enjoy to travel there. So that's for a bit of the background. Then if I go a little bit more into the career path. So in terms of education in Quebec City, I did my bachelor in microbiology. That's already quite some time ago, around 2004 or so that I started there. My studies there focused on virology. Here you can see on the left, I think you can see my pointer probably. We have bacteriophage, so these are viruses that infect bacteria. I spent quite some time in a lab which had a lot of collaborations with lactic industries. So cheese producing, yogurt producing, and those viruses actually cause problems because they can stop the production of acid, so they block the fermentation process, and therefore that prevents from reaching the final stage of the product. Interestingly, in that lab, that's also the lab that discovered the CRISPRs. So you might be aware of that revolution in genetic engineering. So CRISPR was originally discovered as a collaboration between that lab and Danone when they were studying something that has been coined the immune system of bacteria. And then it evolved into further research in the US and abroad as well, to then the use of CRISPR and how to hijack this mechanism for genetic engineering. So that has been a very interesting time in that lab that now made also the CRISPR renowned worldwide. Fortunately, I was not involved in that discovery, but that was very interesting to be around it. Following that bachelor, I realized that I had more of a passion towards human health rather than lactic bacteria and industrial productions. So I moved to Lausanne to finish my bachelor studies in biology. Lausanne is a beautiful place next to the lake. It has the Alps nearby. That has been a very interesting memory there, since I also had a lot of time to travel and discover Europe. So that was a very big highlight of my my studies. Despite them career-wise, maybe the knowledge was not the biggest learnings there, but discovering Europe was very interesting. Then I went back to Quebec City to continue for my master studies. And then I did the master studies in cellular and molecular biology, where I focused more on cancer-related topics, DNA damage, DNA repair, homologous recombination. So this is what you see illustrated here. I won't go into the details of this, but I've been studying some proteins in there, some homologs of RAD51, which are involved in the repair. It was a lot of biochemistry studies, protein production, purification, looking at interactions with DNA. So that was very different from what I've done before. That brought me much closer to cancer, and I really like this, to study how cancer actually happens, and that opened up my whole passion about studying cancer and trying to fight it. What you see on the picture there, I thought it was nice to share. So this is a very old church, actually, so which fire destroyed, and only the front remained intact, and they converted it in a scientific research center. So all the new part is basically a large extension of labs, and you still enter like in the church, but then you go and do science there. So that was a very nice time there as well, focusing on DNA repair. After my master, then I was looking for places to go do my PhD. There was very good opportunities in Canada. I was more interested to go abroad. So I've looked a bit into the US and different regions. And I also looked in Europe to find nice places. And I landed in Zurich mainly based on the contacts and the network that I established when I was in Lausanne. There I got to know a lot of people that also traveled for their master studies, and a lot of them recommended to go to Zurich. And that's where I went for my PhD, and I stayed ever since. I've been here now 10 years, spent the PhD at the University of Zurich. This is actually here, the building with the green roof in the Zurich city. And then just next to it is the ETH, which is also another very big institution in Switzerland. And then that shares the campus with the hospital, which are the different buildings of the hospitals here. Zurich is a wonderful city. The science is amazing. The technology that is available is also enabling a lot of discoveries. So that makes it a very dynamic system with a lot of talents as well, that is useful for creating biotechs and lively environments in Zurich, and the academic research is also leading many things worldwide. Then following my PhD, I was still interested in, well, I didn't really describe my PhD actually. Maybe I can dive a little bit more into that. So it was cancer immunotherapy, the topic that I'll share with you today as well. And I'm very glad to see that we have knowledgeable people already. And as I understood, you have a little bit all of a background in immunology and medicine as well. So I'll try to touch on those topics, and we'll be glad to discuss with Mushref and Adab as well. So during my PhD, I was studying, well, I did a lot of things, and as maybe some of you know, a PhD can be a challenging time as well. The first 2 years of my PhD, I've been doing maybe 2-3 projects that led to different directions that were not really promising. One of which was engineering a new genetic model, so a new transgenic mouse. I spent a lot of time on this, doing DNA clonings, and in the end, it didn't work out, but the idea was really great, to have a mouse that could have an inducible cancer, which is endogenously growing and not injected or induced. And nowadays, there are many models like this. So this is very valuable tools that are around for cancer immunotherapy research. And then following that little failure, let's say, of that project, I continued for 3 more years on a more promising approach, which then was combination of immunotherapies. So I studied mainly prostate cancer in preclinical models, combining various molecules, mainly antibody-based or cytokines, which can influence the immune response and trying to drive a strong and bring a memory response against cancer in those preclinical models. So that was work that was in line with what Roche has been doing as well during those years. And that's what brought me actually to one of the sites of Roche. This is based in Zurich as well, where then I joined as a postdoctoral fellow to continue studying T cells, how to boost them against cancer, and with all the mechanisms that we can exploit to then generate drugs. So this is really where my passion is, to really continue doing research, push the boundaries towards improving healthcare for cancer patients. And that's what I've been working now for 5 years at Roche. It's quite a nice story actually. On this picture, you can see at the bottom, it's actually a garage with all the tires here. So this was a spin-off of the ETH, so a small biotech that started in, I think, in 2000. And then after 5 years, they had a molecule that was very interesting for Roche. So Roche acquired that biotech. And since then, the site has grown constantly over the years. The first molecule is now in the clinic and saving lives of lymphoma patients. So this is a big achievement for the site. And now we're roughly 200 people on that site to bring new cancer immunotherapies to patients. So the site has grown quite a lot in the past few years. So now we have a beautiful tower. I also grew with the company. So from a postdoc, I rapidly transferred into a scientist position, and the growth has continued. So now I'm leading a small lab. I have 2 employees that are doing a lot of the lab work. I still enjoy a lot doing some lab work myself. So I still go to the lab, perform experiments now and then. And I'm leading a few projects as well within the company. Everything we do at the tower is cancer immunotherapy focused. So that brings me towards explaining a little bit of Roche. I don't know if Mushref or Adab, you have any questions for me on that part before I jump into the company business.
From my point, just extreme jealousy, my friend.
So to explain a little bit Roche, because it's a huge company, right? We are 100,000 employees worldwide. There's a big network of academia and industry partners, so over 150, and probably as we speak, that number is probably greater by now. They interact with multiple units of Roche. We have the early, so the research and early development units, which are 3 centers that are totally independent. There's PRED, which has sites across the world. There's Genentech that you might be aware of in California. So this is one of the huge research unit as well. And there's a site in China for Chugai. Each of these have very innovative ways and their own approaches to research and early development. So we all have different types of molecules that all merges together towards a global and worldwide execution of product development, manufacturing, commercialization. So there are teams in, I don't know how many countries, but that hopefully covers the globe for all these aspects. And then there's also another unit at Roche, which is very considerable. So it's about the diagnostics. So Roche is really a leader in diagnostics as well. And I think now you might have heard on the news, there are also kits that Roche are now providing to fight this pandemic. So we continue to really try to lead in diagnostics, but also to bring new medicine to treat patients in various areas. So the framework within PRED is focusing on 6 therapeutic areas that can be quite broad. We have oncology, which I'm part of. Then there's infectious diseases, inflammation, immunology, neuroscience, rare diseases, and ophthalmology. We have multiple therapeutic platforms, so we can create a very broad spectrum of molecules. So all of these can be applicable to all of the therapeutic areas. I'm focusing on large molecules, so antibody engineering and antibody-based approaches. Historically, we also have a very large contribution into the small molecule world with inhibitors and then RNA therapeutics as well. And in terms of therapeutic platforms, this is always evolving with the research that is being done worldwide. So PRED Oncology has 4 sites across Europe. The one in Zurich that I mentioned, which focuses on cancer immunotherapy. We also have a pharmacology team focusing on in vivo work. We have biomarkers, reverse translational science, so many doctors that are planning phase 1 studies, and then we can chat over coffee to bring the knowledge from the clinical trials back to the lab. There's strategy and operations and external innovation to be sure that we can spot the new best big thing, basically. In Munich, it's a huge site. It's mainly diagnostics. And then we have research as well for oncology, where we have early clinical development, biomarkers, pharmacology as well, external innovation. So there's a lot of overlap and interactions between those 2 sites. In Basel, we have the molecular targeted therapies, which are more focused on the small molecule modalities. And here again, we have the same span of functions there. And in Velvin, we have then strategy and operations mainly. And with this, then I'd like to say that what makes the big difference for Roche is really the passion of the people and the skills that are there, really bringing the teams to work together. And we all share one goal, is to discover and develop breakthrough medicines that one day may have the potential to transform lives. And here's a link if you're interested to hear what people have to say. So we've been taking a stand against cancer recently. So there's a few interviews there that explains a bit in detail what we do. And with this, that would conclude the part for the introduction of myself. To go broad on cancer, I can continue maybe on a few slides as well.
Yes,
but
of course.
please interrupt me if you have any questions. I'd like to make this interactive as well.
Yes, of course. We'll probably midway of, depending on what slides you show us, we'll probably go in and start asking about the immunity and inflammation and immunotherapy. But yes, we'll keep our mics open for this.
So in terms of fighting cancer, there has been, well, the fight has been going on for long, right? This is not a new problem, but there are key pillars in that fight, one of which is prevention. This is not something that I'm involved personally, but I think that has a huge impact. Knowing what causes cancer is very important. There's a lot of research still ongoing on this. For example, for tobacco use, which is one of the major causes of lung cancer, obviously, then obesity comes as the rank number 2 of the top factors. This is all based on US statistics as well. There are cancer-causing pathogens that we're learning more and more. There are still many unknowns on those cancer-causing pathogens. Some are viruses, some are bacteria. The influences, so the interaction between the microbiome and the host can be causing cancer, but can also be beneficial. So that's something that there's a lot of unknowns in there right now.
And it's a new field emerging with the relation of microbiome with nearly everything in the immune system itself. So it's
very exciting.
It's unfolding. Yeah, it's very exciting and very very interesting that we'll try to brush up today.
It's a field that has extreme complexities due to the number and differences of bacteria and across populations. And I'd say the research in general likes to have things that can be applied broadly at first. So like the lower hanging fruits, let's say. Yeah. And I think the microbiome is definitely a promising research path that will lead to some therapies, hopefully in the future. But there's still a lot to be discovered there. And then there's other factors as you see here. So inactivity, diet, UV can cause also a lot of cancers. There's early detection, which really improves constantly. So the diagnostics are making progress every year. There's screenings for certain types of cancers, and the awareness of the population on early symptoms, also to know what to look for. And when all this unfortunately continues towards the disease path, then the therapies come in. And there's a lot of research and development. Cancer is one of the major causes of death worldwide, and therefore there's considerable investments into research and development. And there has been some revolutions lately. So the number of approaches has been growing exponentially with the knowledge that we have on new molecules and the biology of cancer. Here I'd like to show a little bit of the progress that has been made. So you can see between 1975 and 2008, which is in blue, the progress, so the improvement in the 5-year relative survival rates for selected diseases. So you can see overall that the leukemias or the blood cancers have been improving a lot in terms of 5-year survival. So the therapies and the screening and the prevention has had a very big impact there. But there are many indications where there's a huge unmet medical need. The lung cancer, for example, is very hard to treat and has a huge population affected. Pancreatic cancer is the hardest to treat. And so there's really a big unmet need there, and that's why we have so much investment into research and development of new medicines.
Okay, then the question from here, Ali, would be traditional treatments in line with chemotherapy and radiation and others. How have they fared so far, and what are the pros and cons?
So conventional treatments have been breakthroughs that contributed to those improvements. Surgery being incredibly efficacious in treating, I'll just go maybe directly to this one. So surgery is extremely effective, but only for localized tumors at an early stage. And it also has some risks, and well, it is invasive, risk of bleeding or infections following surgeries, and it has a very poor efficacy at treating metastasis. So in certain settings, this might be feasible, but depending on the spread of the disease, then surgery is not always an option. Chemo has been around for a long time, and there are constantly novel ones being discovered as well. It's very effective on localized and metastatic tumors, but it's extremely potent and toxic for healthy cells and tissues. I think you probably all know some side effects from it. There's some images in movies that can easily stick to the memory, like hair loss, or there are many more drastic side effects from chemo, which make it a therapy that is difficult for the patient to go through. That has a heavy burden on the patient, but it's always a risk and benefit comparison, right? For radiotherapy, it's a little bit similar. So it's very effective for localized tumors. It's better tolerated than chemo. It has good effect and it's quite potent at inducing DNA damage, at killing tumor cells, but it also damages the healthy cells that are surrounding it.
Do we know if it's preferred between different types of cancers? So what cancers respond better to chemo? What cancers respond better to radio? Are there preferences in terms of the types of the cancers?
So there it gets really into the decision trees of the medical field, in terms of which cancer type is treated with which therapy. This is not really my comfort zone, but yes, there are definitely preferences. Some therapies are tailored for certain types of cancers. On this, I believe every country has their own guidelines. Therefore, the treatments might vary slightly between countries. And
Okay.
there, I can't provide, let's say, a broad statement that would apply to everything.
Okay, so if we would then take this to the topic of today, the immunotherapy, it's not really a new concept, is it? Even though it's been practiced for fairly shorter years than the conventional therapy.
The history of cancer immunotherapy is very interesting. I believe it started in the early 1900s, around Einstein times. There was one doctor, I think, Coley.
William Coley, yes.
Yeah, he was one of the pioneers into cancer immunotherapy, despite the lack of understanding of the biology at that time. They were using, for example, bacterias or inactivated bacterias that were injected and saw some efficacy in some cases. So that started this way, and then the research followed on this. A lot of hypotheses were made. Unfortunately, the models were not available to study properly the mechanisms and to understand it better. And it took a very long time to then trigger this belief that cancer immunotherapy has a huge potential. There were some events during that century that really slowed down the research on cancer immunotherapy. And that led the effort much more towards chemotherapy, for example, and radiotherapy.
How would we describe immunotherapy? What is immunotherapy?
How to describe it?
As in definition for the audience.
As a definition, one could refer to the dictionary to have a proper one, and mine would be really to anything that supports tumor cell killing via the host immune system.
So utilizing the immune response, facilitating it, directing it, maybe manipulating it to attack an invasive cancer.
Exactly. So everything you mentioned is part of cancer immunotherapy. And this has, for invasive cancer, but also for localized tumor, can have efficacy too.
And we'll see today in our discussion. We'll see today in our discussion that it showed varied results between solid and non-solid types of cancers and types of tumors. Okay, Ahdab, you've got a point you wanted to present.
Yes, maybe the definition that I like to say about immunotherapy is just harnessing the immune system against cancer, using these powerful immune system components, including cells, proteins, and effector molecules to target cancer after you understand the mechanism or how the cancer grows, what are the molecules that are using to grow, to invade the immune system. So, and also I would like to add to what Ali said about surgery, chemotherapy, and radiotherapy. Although we do have the immunotherapy has shown a very promising result, but now most of the successful treatment is a combination between traditional and immunotherapy. Like you see, for example, immunotherapy plus chemotherapy, right?
Yes, very interesting point, and I think it's very important to say at this stage of the lecture. So immunity now, immunity and inflammation and cancer. So we're trying to manipulate the immune system or harness, using a better phrase from Ahdab, harness the immune system to fight cancer. What can you tell us about that? We know it's essential to understand how a cancer interacts with the immune system. And our understanding then generates or leads to a term cancer immunologists use as immune editing. What can you tell us about immune editing, Ali?
So about immune editing, or to step back maybe one step before this, it's really important, just like Ahdab mentioned, that we have a good understanding of the disease biology. I can just go back a few slides. And what makes cancer so difficult to treat is that first, it has a genetic component that can be either inherited in some cases or acquired through mutagenesis. And this is polygenetic. There are very different mutations that can drive cancer progression. This is an unstable process. Cancer is very heterogeneous. So you have within the same tumor, you have a number of cells that all will have different mutations, they will share some mutations, but not all. So it's a little bit of a game of survival of the fittest, if we can say that. And that once it's spread, then it's much harder to treat as well.
And does that give each tumor its distinctive marker then, these different mutations and protein productions?
So the mutation is one layer. There have been some mutations are very useful in therapies because they can be used as biomarkers. So to distinguish a certain type of tumor and to have a biomarker that we know we have a therapy that will work. So there
Yes,
are
I do
therapies
have it,
that are specific for some mutations, but this is not always the case.
Yes, I do agree. Always the mutation burden is very important to classify the tumor, whether it is less immune. For example, the treatment works better with the highly immunogenic tumor, basically that tumor that have a high mutation rate compared to the tumor that have low mutation rates. So yeah, like for example, pancreatic cancer, do you want to explain that maybe?
Yes, of course.
Am I right, Ali? Yeah.
Yes, you are totally right. So I was coming from the side of the mutations towards the therapies, more in terms of small molecules that I was thinking.
Great.
But what you mentioned is totally right in terms of cancer immunotherapy. The higher the mutational burden a tumor has, the higher the chances are that the immune system will recognize it as aberrant.
And it recognizes it because what exactly? That's what I'm trying to lead to. What in mutations makes it recognizable?
So in the cancer immunoediting process, there are a lot of various players and various mechanisms. The important one would be the presentation on MHC molecules, where then you have tumor antigens that can be presented. These can be peptides that contain mutations. And I think that's what Adab was mentioning, that the higher the mutational burden you have, then the higher the likelihood to have peptides here presented that can be recognized by T cells and then eliminate the tumor cells. This is one part of the cancer immunoediting. So this is one that is tumor antigen dependent. You also have editing that occurs via other molecules or other effector cells. For example, the NK cells, you have macrophages, you have other types of T cells that are not necessarily recognizing mutated peptides, but other types of ligands, and dendritic cells that present a mixture of danger signals. All this leads to elimination, hopefully, of the aberrant cells. And then this is part of the successful immunoediting or the successful elimination of the tumor cells. The immunoediting consists of the path where you go towards an equilibrium, where you have the overall or the net outcome is that the tumor cell death is basically equal to the tumor cell growth. So it stays in a rather stable state. And during this time, you have a selection for cells that will evade the immune system. And if some cells, for example, lose an antigen that was presented and that used to be recognized and lead to tumor cell death, if it loses that protein or antigen, then it manages the escape. And that's where the danger is of cancer progression.
So it's less of a failing of immune cells, more of the evasion and the survival mechanisms of tumor cells.
So I think the tumor has a lot of mechanisms intrinsically to overcome the immune system. Both can occur. You can have a failure of the immune system due to other therapies or some immunosuppression or other disease that lowers the immune system, and then that will favor the balance towards the escape. You have tumor intrinsic mechanisms where then they will secrete factors that prevent effector cells, so the T cells will be inhibited. For example, we mentioned earlier that IL-12 is a very potent cytokine that favors the T cells to kill the tumors and to maintain a phenotype or an active phenotype to continuously kill. There are tumors or regulatory T cells that can secrete other factors that then has the opposing effect. So on this, it's always a balance in terms of the tumor environment, and the tumor cells always try to evolve and to escape this. That can be through antigen loss, MHC loss. So if you lose components of the machinery of the MHC, because those peptides are presented via machinery that requires a lot of proteins. So you have processing, degradation of entire proteins to make smaller segments. And then those peptides are loaded on the MHC. So all those proteins can end up with mutations, and then that can alter the presentation mechanisms.
So it appears that what I understand of immune editing, it's a process, it's a dual process where the immune response can suppress and also promote cancer growth.
Exactly. So it's really a double-edged sword. You have some cells that are, let's say, constantly on the good side. There are some cell types that play on both sides depending on the context and the signals that they receive. And there are some cells that are usually detrimental.
So we know of studies or a series of reviews that came out from the Journal of Clinical Investigation. Tang colleagues wrote a very remarkable review on CD8 positive T cells and their role in immunoediting. So the CD8 role in immunoediting, can you tell us a little bit about that, Ali?
Of course. So the CD8 play a major role in the killing. They're really the most potent tumor cell killers that we know of, I believe. And there's a lot that can be done to support them. They require very specific signals that can be, so let's say 3 main signals. You have first the T cell receptor signaling that activates the T cell. This is an interaction between the T cell and the dendritic cell that is presenting an antigen. You have the signal 2, which is then co-stimulation. Co-stimulation is quite complex. It's a lot of different proteins that will be interacting on the dendritic cell and the T cell, so they will form bridges. And all the pairs, some have a positive impact and some have a negative impact. And the balance will decide of the fate of the T cell. If you have a good gas pedal, let's say, then the T cell will have a positive signal and then will be activated much more efficiently. And then there's a signal tree that will come in with the cytokines that provides a feedback loop as well, so that the T cells differentiates, proliferates, and becomes a strong effector cell. On the opposite, you have some downregulation that happens on the signal 2, some of which are written here. So PD-1 and CTLA-4 are now very well characterized to lower that signal 2, and that reduces the the positive signal. So it really blocks the positive signal, and then the T cell will not be able to differentiate, proliferate, and be a strong killer.
It's important that you've mentioned that, because we'll be talking about this in a few minutes regarding the immunotherapy with checkpoints, immune checkpoints. Inflammation in general can play also dual roles. So chronic inflammation promotes enlarged tumor survival, cancer survival, progression, while acute inflammation tends to do the opposite. I'd like to take you now to the microenvironment of the tumor and how that facilitates the way the tumor survives. So there's no doubt now that tumor infiltrating lymphocytes are present within tumors. While they are inactive in vivo, in vitro in the laboratory environment, they are sufficiently active. They are working enough in a sense where they can compete and defeat tumor cells. So it appears that within the tumor environment, there are elements that we are yet to understand. What can you tell us about the main three elements of tumor survival within the microenvironment of the tumor? Reduce of antigenicity, loss of immunogenicity, and the immunosuppressive microenvironment elements, like the presence of maybe an increase in cytokines that are known to have suppressive effects, as in IL-10 and TGF-beta.
That's a very good question. Quite a complicated one too to answer.
It's for you to break down, Ali.
Yeah.
With the help of Ahdab, of course.
in terms of factors, so you mentioned three. I believe there's much more than three. And I think just on this slide, you can actually see many of them that contribute to the tumor microenvironment. You'll have cells, like the MDSCs, so myeloid-derived suppressor cells, that will negatively impact the T cell response. You have Tregs as well. You have secreted molecules that can be pro-inflammatory, as listed here, or inhibitory, as listed there. And all these come together at different concentrations, and the cells have different interactions based on their localization within the tumor. So that has a big impact. One challenge is that we don't have yet the resolution in a 3D space within a tumor to see all of those factors at play. And in the end, the net outcome is then an addition of all of these. Everything needs to be factored in.
I was saying that maybe if we can classify it as like a blood cancer and the solid, like the liquid cancer and the solid tumors, we do have these challenging, or maybe the local mechanism of immune suppression in solid tumors, because it's a heterogeneous and a mix of many cells, as Ali mentioned. Regulatory T cells, myeloid-derived suppressor cells, and also the effector cells like CD8 and CD4 and the other, and antigen-presenting cells. So the solid tumors, or maybe we can say the tumor microenvironment, is really remain challenging, because there are many resistant mechanisms are going on. It's not only one. I'll give just one example. For example, in the melanoma cancer, it has been shown that 3 resistant mechanism is actively going, is like infiltrating of regulatory T cells, for example, which is a suppressor T cells, and also the upregulation of PD-L1, which is the checkpoint blockade they were talking about earlier.
So how are they recruited? So that's one thing I've noticed, and this is a question for both Ali and Ahdab. How does a tumor cell recruit these cells? Are there chemokines being secreted? Do we see CXCL9? Do we see other chemokines being let out for these, for different immune mediators and immune cells to arrive in the tumor?
Yes, of course. I think there are many different suppressor mechanisms that the tumor is using to suppress. Like, for example, let's say when you are adoptively transferring T cells, let's say, okay, I'm taking in the setting of adoptive transfer T cells, which is, for example, CD8 that are specific for tumor, they will infiltrate the tumor, and upon active or acute inflammation, this will release a negative feedback to the tumor. So I don't know how to explain it in a very simple way. Maybe Ali can help me.
Is that what you're saying?
Yeah, there is a negative feedback upon the effector CD8 T cells infiltrating the tumor.
Yes, so this is totally true.
So PD-L1 will be upregulated. Yes, Ali?
These are natural breaks of the immune system. So basically, when you have an immune response, being cancer, infection, any immune response, when it drives a strong influx of T cells and inflammation locally, there's an acute phase, and then there's a resolution of the infection or the response. Part of that resolution, there are the mechanisms that were just mentioned. So the Tregs come in to lower the inflammation, to avoid too much damage to the tissue. PD-1 and CTLA-4 are other breaks as well that lowers the T cell. So when the T cells are activated to their maximum effector function, they start upregulating those breaks. So the breaks are present, and then when the interaction with the partner, so the ligand of PD-L1 and the ligand to CTLA-4, then you have this bridge, and that leads to a negative signal into the T cell, so that the T cell calms down a little bit, and it stops secreting cytokines, and then it lowers the inflammation and leads to less killing. So this is an intrinsic protection mechanism that evolution brought us to protect us from too much damage and autoimmunity.
So my understanding of this immunity, Ali, is that within the cancer microenvironment, it's promoted earlier or promoted untimely to push cells, immune cells, to apoptosis. Is that, am I phrasing that correctly?
Yes. So the tumors, not all, but many will develop those, will express those ligands, for example, PD-L1, so that when the T cell comes in, it doesn't really get the chance to do its job. It gets on the brakes right away.
Because activated T cells already express PD-1 on its surface, as you were saying, as part of the natural process of immune tolerance to facilitate the suppression of the immune response. So what you just mentioned, and on the heel of what Ahdab was just saying, I think we should go into one of the first...
I think I just want to add about the PD-1. As you said, okay, it's naturally expressed on T cells, but PD-1 is not constitutively expressed on T cells. Like upon T cell activation, the PD-1 is transiently expressed as an activation marker, but then it will downregulate it. And then toward the effective function, when the T cells started to be exhausted, the PD-1 will be expressed as an exhaustion marker. So, and back to the point that about the CD8, I think the emergence of the immune suppressive pathway in the tumor microenvironment is triggered. Maybe we can say it in a better definition, is triggered by inflamed CD8 T cells in the tumor microenvironment as a negative feedback mechanism. So I just wanted to add on this and this on PD-1.
So we're going to talk. Go on, Ali. Sorry.
Sorry, I was just gonna say that I totally agree. So each break on the cells, they have different kinetics over time, and that's what makes it so complicated. And the inflammation drives the adaptive resistance mechanisms within the tumor. So if you have a strong inflammation, a lot of interferon gamma, for example, that will upregulate the PD-L1, which then will support the break on the T cell. So there's so many mechanisms that contribute to it that it's difficult.
Yes, exactly.
It's really a kinetic and going up and down and it's multiple proteins and soluble factors contributing to it. There are also tumors that are kind of ready, that they readily express PD-L1 without interferon gamma, for example. And that is another layer. Yeah.
So on the heel of that, we'll take it to the CTLA-4 and PD-1 point. You've elaborated there with the help of Ahdab on how CTLA-4 and PD-1 is used within the tumor microenvironment to promote tumor survival. So we know that CTLA-4 is expressed on CD4, CD137, CD25 activated T cells. So it is part of your natural immune tolerogenic effects or processes. It binds to the CD8086 receptor on dendritic cells, macrophages, and B cell, which are activated as well. PD-1, as Ahdab highlighted, is not only solely expressed on T cells, it's on T cells, less knowingly on B cells and NK cells. So we don't know really a lot about what it does there. Not surprisingly, if it did the same function. And it has two ligands, 1 and 2, which are found on macrophages and dendritic cells. It's a programmed cell death protein, facilitates apoptosis through BATF, which counter-affects transcriptional progression in the nucleus. So quite prominent roles for PD-1 and CTLA-4 in the immune response. And now they've been targeted in immunotherapy. What can you tell us about that, Ali?
Okay, so let me change a few slides forward. In terms of checkpoint blockade, so PD-1 and CTLA-4 are called checkpoints. So they're really breaks for the immune system. I think here we've discussed already most of those points. There's really a tightly regulated balance, and there are many opposing co-stimulatory receptors as well. So the two major breaks that are known and that have shown clinical importance are CTLA-4 and PD-1 or L1. So this is, at the moment, I don't know how many antibodies are actually existing and being tested in the clinics for these pathways. There are some major ones that are known. I've listed a few references down here. Interestingly, the Nobel Prizes last year, Physiology and Medicine, are about this. So that's very interesting read. And they have shown clinical relevance. So they have those molecules, so the antibodies that block those pathways can inhibit the negative signal. And that saves lives, basically, in terms of cancer patients.
And is it specific for T cells, Ali? Because as Ahdab was saying, and as the literature proves, while they are mainly expressed on T cells, but they are found on other molecules, other cells, how can we determine specificity for T cells, or is that not important in this frame?
So for CTLA-4, I believe it is mainly, so the research has really been focusing on T cells.
Yeah.
For the PD-1 and L1 axis, there's new data that was published, I believe, in the past 2-3 years, showing that the blockade of PD-1 or PD-L1 has an impact also on the antigen presenting cells. So that provides different mechanisms of action, which all in the end lead towards a better T cell killing of tumor cells.
Okay.
I think there's still many unknowns. There's still a lot of research being done on the mechanisms to see how broad are the impacts of a blockade on such molecules.
And let me take you to one of the most interesting parts of immunotherapy, the chimeric antigen receptors on T cells. T-cell immunotherapy. It's, I mean, the least you can say about it, it's fascinating. It's been in work in the last 10 years, I think, while being discussed much earlier. What can you tell us about the chimeric antigen receptors, or CAR T cells?
I totally share the excitement with you. I can list a few of the cancer immunotherapies type.
Yes.
So after the checkpoint blockade, these are the ones that really laid the foundation for cancer immunotherapy to really expand and bring a lot of research in that direction. And then that favored the discoveries across the board in many different approaches. T-cell therapy or CAR T-cell and adaptive transfers, there are many different ways to do that. It started, well, let's say the first major successes were T-cell therapy based. We're focusing on using the T-cells that the patient already has. So isolating the T-cells from the tumors, expanding them, activating them, and reinfusing them to the patient in huge amounts.
So when you say from the tumors, are you talking already infiltrating the tumors?
Exactly. So really taken from the biopsy of the patient, isolated in the lab, and then really activated and expanded. So if they isolated a few thousand cells, then they could go up to a few billion cells, and then reinfuse this to the patient in the hope that these would be tumor-specific T cells.
Okay, so I have a question about that. These cells are already programmed; they've already been activated and you know, specified. Why don't I go to the lymph nodes, for example, where you've got naive T cells there, where you can genetically modify them? Why tumor infiltrating cells?
So in that first approaches, they were not engineering them. So these are really the natural receptors of the T cells.
Okay.
And then taking them from the periphery or from the lymph nodes, you would probably have some cells that should not be activated and expanded at the risk of autoimmunity.
Okay.
I think that's from this part, and then that gave a lot of hope to engineer and really hijack this by putting a T-cell receptor of desired specificity, and that brought then the CAR T-cells. The CAR T-cell is a booming field. There has been some successes in the clinics already and some approved drugs, mainly in hematology cancers. The CAR, so the chimeric antigen receptor, is a construct which is a fusion of, for example, an antibody, so just the binding domain of the antibody. Then there's a transmembrane domain to anchor it on the cell surface.
T-cell surface.
On the T-cell surface. That comes from a totally different protein. And then you have the chimeric part, which are the signaling domains. And there, they have been, so there's multiple generations of CARs. You have the first generations contain standard T-cell receptor activation domains, like the CD3 zeta, for example. And now the new generations combine this with multiple other signaling domains for co-stimulation. So with a single receptor, you can basically cover signal 1 and 2.
I see. Do you still need antigen presentation?
With this, you don't. That's the beauty of it. You basically bypass the whole mechanism, and then you rely on your antibody binding. So as long as you have a good target, then that will lead to quite a potent T cell killing.
That is fascinating. So you got a T cell that binds directly to the antigen, or the tumorigenic antigen for this case, and it reacts directly. It does not wait or does not go through the process of antigen presentation, antigen recognition first, second, and then cytokine stimulation of signals. So it responds straight away. My first question, what type of T cells are they? CD8, CD4?
I believe that most approaches are using CD8 mainly.
For cytotoxic abilities.
Exactly, as they are more potent for killing. However, the CD4s that provide the T cell help is very beneficial, or has been shown in multiple other settings where the CD4 help provides secreted molecules that support the CD8 effector functions. So I think, I'm not sure there, but I believe that there are some constructs that are being used in CD4s and CD8, and then reinfused to patients. These haven't made it yet through all the clinical phases, but this is something that has a high potency, and that also brings some risks associated with it.
Hadab, what do you know about this?
So I do agree with Ali. The CAR T cell therapy concept is very, very beautiful and very intelligent to do a synthetic receptor that combine the antibody affinity, because we know that the antibody affinity is higher than the TCR, for example. So the idea of the CAR T cell therapy came from that things, because most of the tumor antigens have a lower affinity, especially if they are a tumor associated antigens, so they are expressed at a low level on the cell surface. So so I just want to highlight another type of so basically the CAR T cell therapy, we are here redirecting the specificity of T cells using CAR receptor, the chimeric antigen receptor. Before the CAR, we had another type of, to redirect the T cell specificity using the TCR, which is the natural receptor on the T cells. So we are just finding a good target to tumor antigens, and we introducing this TCR into the T cells using a genetic engineering tool. But the problem or the limitation with the TCR, as you said, is we need the MHC presentation to present the antigen through the MHC by the antigen presenting cells. Unlike TCR, CAR has an antibody-like specificity capable of activating the T cells in an MHC or HLA independent fashion. So I just want to highlight the other type of and how to redirect the specificity of T cells, especially that I was working on during my PhD. That's it.
Can I ask one more question about the CAR T cells?
Of course.
How long do they last in the system? And can there be memory cells from these T cells?
That's a very good question. I think in terms of persistence, this depends a lot on the construct that is being used. Depending on which generation of CARs that is. So if you have a CAR that has a strong signal 2 as well, that will most likely provide a longer persistence. In terms of memory, I'm not sure actually what is the longest time that those cells have been observed, but I know that they can stay very long.
What's very long? I don't want to put you on the spot.
I think well, I would have to go back to the literature a little bit, but I believe we're talking months.
And is it a one-off treatment, Ali? This is from a complete novice. I don't know how this is done. Do you know if it's a one-off treatment with CARs where you depend then on in vivo proliferation?
I know that the once the CAR is infused into the patient, they do expand as well when they have when they recognize their target. I think so. The production of such therapies is so complex to manufacture. I believe it's a one-off, but I'm not sure. I think there's still new approaches that are being done in the research and early development stage, where then they look at constructs that don't necessarily depend on the patient's T cells, but just at a pooled, at donors, basically, so that you have an allo transfer.
And do you, so I'm guessing it works with donors because T cells don't express MHC. So that's a MHC2 at least. So is that why it's you can use it from pooled T cells rather than from the patient's T cell itself?
I'm not sure there how the reaction between the host and the transferred cells are. I think that would probably impact the persistence quite strongly. Even if the transferred cells don't rely on the MHC mechanistics, you do get an allo reaction usually. So if you get infused cells from somebody else, then that usually come with immunosuppressants as well. But I'm not familiar with the protocols in the clinics for these therapies.
Okay, I've got.
I just know it's still very early days on that, and there's a lot being done on future therapies for this.
So immunotherapy, and for patients who are already being treated with immune suppressants, so autoimmune patients, how does that, is it better than the conventional chemotherapy and radiotherapy? Because it's less broad and more targeted. Do they fare much better?
So I don't have concrete examples to provide a comparison. So to say that something is better than another therapy, that really then belongs into the doctor's
MDs.
Yeah, into the MD's field and decisions for, like, they know what's best for the patient. In terms of potency, and so there's pro and cons at each approaches, right? The T cell transfers, when they're engineered with a new T cell receptor or with a CAR, it provides an extremely potent killing function. But this has been also linked to some toxicities. So if it's too potent, that can have some safety risks. So it's always a window, the therapeutic window is the key here for every therapies that are used. So they always come with some safety risks. And with those CARs and TCRs, the specificity is key. So if you have a specificity to a target that is also expressed on some healthy tissues, then that is a big risk because it's extremely potent therapy. If the target is very clean and expressed only on the tumor cells, then that approach is very, very strong, and that's illustrated by the two references down here. In blood cancers, you have targets that are very specific to the B cells, for example. So that makes it a very valuable approach there. If we go in patients that are immunosuppressed for XYZ reasons, then you will want to have a direct killing on the tumor cell. So if it comes from T cells that are infused, or from radiotherapy or chemotherapy or other approaches, having the direct impact will be very valuable.
So on the heel of that, it's shown most effective, or I mean, I don't know if you can, maybe on better terms, it's shown some good results in B-cell lymphomas. So targeting CD19. Do we know why it's more effective in lymphomas compared to solid tumors?
I think there it's maybe a personal opinion. I think that's mainly due to the target expression and how specific the target is for your target cells. In solid tumors, it's a little bit more tricky to find a target that is not expressed elsewhere.
Okay, so it's down to the B cell being well defined and the CD19 being a good target. That's why it's a more efficient approach. I
think this is one of the major challenges that the adaptive T cell transfers will face or are currently facing.
Ahdab, do you have any input?
Yes, I can add to the CD19 CAR T cell. I think it's shown a good result as well, because CD19, as you mentioned, it's expressed only on B cells. So when the B cells are removed, because we still have a disadvantage of removing all the normal B cells, not only the malignant B cells from the circulation, but here the patients can have the temporary adoptive transfer of antibody until his normal B cells, his stem cells producing the new B cells and release it to the circulation. So this is what I would like to add with
That's a very good point. Yeah, very good point. That the transfer of antibodies can really protect the patient as well when there's a strong B cell depletion induced by the therapy. Yeah.
So I have, I'm going to raise one final point, and then we will open the panel for questions. So I'd like to remind everybody to please, be brave, raise your hand, ask your question, or if you have a comment, something you want to add, just please raise your hand, and we're more than happy to hear. We've got plenty of time, and Ali said he's free until tomorrow morning, so that's absolutely fine.
Yeah, it's weekend.
Yes. Maybe a stretch of your scientific knowledge, Ali. It's quite new. Microbiome and cancer immunotherapy. So, I read a really nice paper last year talking about the effect of bacterial fatty acid chain products like butyrate on the secretion and the activation of CD8 memory T cells, for example. So what I'm trying to say here is we're seeing more and more the effect of bacterial products from the bacterial population in our guts and different parts of the body. Being that we see now how much effect these microbiomes carry on different systems in the body, how much of an effect does it carry in cellular immunotherapy when we're trying to manipulate or harness the immune response?
I think that's a very interesting question. At the moment, we're still trying to sort out the complexities of it globally. So the microbiome is very complex. I don't know exactly the number of bacteria that every human has, but it's a huge amount. And the interaction that this has with the host immune system is still poorly understood. There are some very interesting papers on this, as you mentioned. It also has a double-edged sword, basically. So you have some bacteria that seem to be beneficial. The current studies have been trying to identify these and to see if we could just provide those bacteria to the patients. The results are a little bit I wouldn't say conflicting, but there it really seemed to depend on which populations you study. So for example, a European study or somewhere in America, they might, or there's some papers that have identified some bacteria that seem to have a positive effect, but they're not always the same. There doesn't seem to be a good overlap, and we don't really understand why. So that is one part of it. I think it's just that we don't understand yet sufficiently to be able to use that as a very good approach. I think there are some interesting papers in the use of antibiotics as well, and cancer therapies, and the links that this might have. These are very interesting reads if you're curious into that topic. I'm by far not an expert into this, but this is something that, yeah, I think in the next few years, then once we understand it better, there might be some ideas on how to use this for patients.
Okay. All right. Well, thank you, Ali, for the lovely discussion we had together. Ahdab, how are the audience looking?
There are some questions in the chat, and some people are interested to ask a question. They raised their hand. So which one shall we take first? Let's take one from the chat.
Okay.
Okay, there is a question about the gamma delta T cell. They ask, what is the role of gamma delta T cell?
Do we know who asked the question?
Yeah, it's Muhammad Al-Qasim.
I
think it's quite general question. He's asking what is the role of, because we know, for example, like CD8 is very efficient anti-tumor immune cells. So what about the gamma delta T cells?
Well, thank you very much for the question. The gamma delta T cells are quite different to the standard alpha beta T cell. So the CD8 and CD4s, the conventional ones that we talked about, have a TCR that is composed of an alpha and beta chain. The gamma delta have a TCR composed of gamma delta chains, and these bring a different specificity. These cells tend to recognize molecules in a different context, so not necessarily MHC presented, and they recognize modified molecules like peptides, like that can be I don't know, phosphorylated, for example, or aberrant molecules. And these T cells are usually more tissue resident. So you'll have, for example, in the gut, a specific gamma delta T cell population. They're usually talked in terms of numbers based on their gamma delta chains. So I forgot exactly which numbers that is, but I believe, I think it's gamma 9 delta 2 that are in the gut. And you have, in different tissues, you'll have different types of gamma delta T cells. And when they sense something abnormal, then they can trigger a very rapid inflammation. And the CD8 and CD4s that are alpha beta are more circulating. So they're scanning for signals to be instructed to kill or instructed to just continue passing by.
So I guess I see the logic behind his question, because, well, more or less, of course, with huge variations, you have already a subset of T cells that react directly. So maybe that's what he's trying to say. Isn't it easier to engineer them compared to conventional T cells, CD4 and CD8 T cells?
If it's easier to engineer them, that's a good question. I know that there are efforts being done. There's some research and early developments on those cells as well. The fact that they represent a very small fraction of the T cells in circulation, makes it a little bit more difficult to work with them, since you cannot get large numbers from blood of donors.
Of course, we'll get a shot.
That is one thing that has slowed down the gamma delta field a little bit, but I believe there are CAR approaches with these cells as well. So I think that's something that will be interesting to read as well in the future.
And that's a very good question, I must say. Very good question. Who else do we have that up?
Maybe we can take Safa. Safa, do you hear us?
Safa. Can you hear us, Safa?
Yes, hi. I'm Safa Turkistani. I got my PhD in the breast cancer, specifically the triple negative. And I'm also a medical microbiologist. I just want to comment about the microbiome and the cancer that you mentioned, Dr. Mushrif.
Yes.
From my point of view, I see that there's always a question, who came first, the cancer or the microbiome? Always the person, the microbiome signature of a person determine the and can help us to predict the outcome of the cancer, because the microenvironment that is occurring in the tumor with the microbiome and their mediators that are like the waste byproducts that is generated by the bacteria and also by the cells, they all are interacting and making long-term or chronic inflammation, which will lead to by the end for a cancer. As we know, these mediators can harm the cells and the DNA replication and the damage and structure, making a lot of mutations. This will lead to finally to a cancer.
Mm-hmm. Mm-hmm. It's a very interesting point.
Yes.
You were there's a point you were mentioning about the types of CAR cells.
Oh yeah. So maybe Dr. Ali can correct me. These T cells that have been isolated from the cancer patients, they are called energetic T cells. By somehow these T cells go inactive. They were active like. I don't know the mechanism of that being inactivated or energetic. They are isolated because they are express specific markers that I have read in one paper, and then reinfused to the patient, so they will become active again. Like they are not educating them in the lab or in petri dishes for that specific antigen of that specific cancer. So they already know.
So you're saying that they were energetic in the tumor?
Yes, functionally
energetic in the tumor. They're
yes.
In the laboratory, they are activated, then reintroduced.
Right.
So okay, so that I think that follows on the same chronological understanding of what we were saying, Ali, where the T cell infiltrates, but in the tumor itself, infiltrating T cells are inactive, but active in the lab environment. So it appears, according to what Dr. Safa was saying, that these energetic cells are then reactivated maybe in the lab. I don't know how much of that you can tell us about.
Yes, of course. Thank you very much for that question.
Thank you, Safa.
So in terms of energy, or depending how people like to call them, the T cells that don't manage to do the tumor cell killing, and that are, for example, isolated and expanded in the lab, I think that there's a big part of it that goes to the signals 1, 2, and 3 that I've been mentioning earlier. Within the tumor, the microenvironment will have, they will not be able to provide the signals 2 and 3 properly to stimulate or to maintain that effector function. And on top of that, there are many negative mechanisms within the tumor. So once the cells are taken out of there and in the lab they are reactivated, they are provided with an optimal signal 1, 2, and 3 at a strength that, and in numbers, that provides the cells to reinfiltrate the tumors, and then to overcome that lack of signals, since they've been instructed already with very strong signals 1, 2, and 3 ex vivo. So I think there's still the challenge that the tumor will still have signals to break those cells, to slow them down. But the numbers and the strength by which they're activated might be able to overcome this in certain cases.
It presents an extraordinary risk there, because inevitably you'll need to change the microenvironment in the tumor as well, because there's a huge influence to drive the immune response down, either by the presence of infiltrating Tregs, TGF-beta, IL-10. So eventually, I think, and this also goes down to my earlier question, how much they proliferate these CAR cells, these genetically modified cells, what's their rate of proliferation, their expansion. They will eventually fall short, or they will eventually succumb, I think, maybe, to this suppressive microenvironment. So I think that's a challenge. I don't know if you agree with me, Ali.
I totally agree that it is a challenge, and it's very hard to make a generalization. I think every patient will have a different response to it. I think there's a lot that can be done. I think you really mentioned a very important point in the tumor microenvironment. We need to reprogram it somehow. And there's immense efforts on this. And the microenvironment can be reprogrammed in many ways. We can see it in terms of classifications of tumors. You have tumors that are already inflamed, or that contains already a lot of T cells at baseline. You have, on the opposite spectrum, tumors that are classified as immune deserts, which don't have T cells, but have other immune cells that somehow prevent them from getting in. And you have a bit of a middle picture, which are the excluded phenotypes, where then the T cells are reaching towards the tumor, but they're just staying surrounding it. And all this has to do with the tumor microenvironment and the signals that are in there, the chemokines, the cytokines that are around, and the cells. Having different modalities, so if one can target a cytokine within a tumor, then that can create a spark to then bring the T cells in. You have other antibodies that can do this as well, that can bridge, for example, the tumor antigen and CD3 on the T cell. And that brings the inflammation up. And that, in turn, if you have a lot of T cell infiltrate and a bit of activity there, that will influence the cytokines and chemokines and the recruitments of other cells. So what's important is really that spark to kind of start the fire there. And then therapies and immunotherapies have better success when the tumors are inflamed.
On the heel of that, I advise everybody to go and read a nice paper by Gregory, Betty, and Whitney Glandley in the Clinical Cancer Research, which talks more about the tumor microenvironment, which I don't think we discussed it enough, but due to the limitations of time. Hadab, are you with us?
Yes, I am.
So what are we doing?
So actually, I do have a question for you, Ali.
Mm-hmm.
So in 2019, last year, a lot of advances with immunotherapy have been made, focusing specifically, for example, on immune checkpoint blockade, like anti-PD-1, anti-PD-L1, and anti-CTLA-4. But with the hard-to-treat types of cancer, like, for example, glioblastoma or any other cancer, do you think, because especially you said that it's important to understand the cancer and the immune resistant mechanism going on. So do you know any antibody that targeting other cells in the tumor microenvironment rather than the T cells, like for example, myeloid cells?
That's a very good question. Yes, targeting other cells is a possibility. I believe in the clinics there are some possibilities there. I'm clearly T-cell biased, as you might have noticed. In the myeloid field, there it's also a little bit complex because those cells will play on both sides depending on the microenvironment. The therapies so far that I know of, I think they aim to prevent their recruitment within the tumor. So to try to reduce, for example, the myeloid derived suppressor cell, to reduce their counts in the tumor. So to have a, to favor the T cell ratio to myeloid suppressors. Other therapies there, we have, if we think of, so we classify things into engagers, generators, and co-stimulators. If we look at engagers, these will be molecules that will bridge with the T cells to lead to a direct killing. The generators are the class of molecules that will trigger a little bit more towards the myeloid as well, because the generators want really to generate a full immune response. So to provide the good signals so that the T cell and the antigen presenting cells talk to each other, educate each other on, okay, what's the target? And yes, you have the green light to go with good co-stimulation. And then the co-stimulators will support what the generators are doing. So there are many generators also being studied at the moment. One key molecule in that is CD40 and the CD40 ligand. These have a very strong impact on how the T cell respond. So these are also part of studies going along the myeloid, the use of myeloid in cancer therapy, but eventually it all goes down to the T cell.
Can I ask a very basic question, Ali, in terms of, have we tried to reprogram T cell recognition by introducing tumorigenic antigens to T cells in the lab and then reintroducing them back in?
So there's been a lot of developments there, mainly enabling techniques. So there's new methods that enable the isolation of single T cells. So taking a tumor, then you can sort the T cells separately, and you can identify their specificity, at the same time sequencing their T cell receptor. And with this, then you can either create a new construct, so your favorite T cell receptor, that you can then provide to the T cells of the patient.
Yes.
So I think this is a little bit what you mentioned, right?
Yes, on the line. So because it makes for, I mean, it is a basic line of immunology, but then you can see how it will impact future immunization as well. That we're not so clear about with CAR T cells.
I think there the challenge is which T cell is the right one. How to define this, and there we have there are a lot of algorithms that are prediction methods to understand what is a good peptide target. Some that are mutated might not necessarily be presented or immunogenic, even if mutated.
I see.
And there, the prediction tools that are currently available, they're not powerful enough. They're missing a lot of let's say of good targets, and the predictive capacity of those algorithms is somewhat biased as well.
Yeah, I think you've answered my question very, very well, actually. So from what I understood, we still don't know what the most prominent targets are, and that's where the struggle is.
And every patient will be different. Every patient has a large different set of mutations. So it makes it a very personalized medicine, which has a lot of advantages. But at the same time, it's very difficult to develop such a medicine in a time frame that the patient needs.
Yeah. Ahdab.
Yes.
So the mic is yours. What do we do?
Yes, maybe let's take a question from Tarfa, Dr. Tarfa. She is one of our colleague and a great immunologist.
Hello.
Hello.
Yes, hello, Tarfa.
Hi, thanks, Ali, for your talk. I'm not a cancer immunotherapy expert, but actually I have a question regarding the DC vaccine, because I'm interested in this point. What's the hope that we have now in personalizing DC vaccine? You know, there's a lot of approaches in vitro and ex vivo, but so far, all of the reviews last year have been highlighting that it's still not successful in vivo. So I just want you to tell me more, if you know, about the development of the use of conventional DC type 1 in fighting, in personalizing a DC vaccine against cancer. What are the hope? What is the development in this? And do we have a specific target, like certain kind of cancers that works with it? And some kinds do not respond well.
Thank you for the question. I think that's a very interesting therapy as well that we didn't discuss much yet in this webinar. So tumor vaccination is also a field that is exploding at the moment, mainly due to our better understanding and the methods we have to sequence the tumor cells and see the mutations that are there. And this can be used to create tumor vaccines. These approaches have been tested for, let's say, continuously. Some of them have worked, but not many. I'm only aware of a single one that is now benefiting patients. That's in prostate cancer.
Okay.
There's the reference written on this slide, and this one is a little bit, let's say, older vaccine therapy. There, they use tumor lysates that can be fed to antigen-presenting cells or dendritic cells and reinfused to generate immunity. And those antigen-presenting cells are modified genetically to express some other proteins as well. It's a highly personalized medicine, as you mentioned. So this is very, so for the patient, that has a lot of benefits there. The challenges are on the specificity for such an approach. There, I think recent progress on the sequencing makes maybe the specificity something that we can overcome. So if we know already which peptides can be used to create a potent vaccine. But again, that goes back to the point that our predictive algorithms for identifying the right peptides, the right targets, this is still a challenge. And then on top of this, then the efficacy of vaccines in terms of tumor vaccination seems to be limited. So the DC vaccines have a lot of promises that didn't deliver on yet. because there are still inhibitory mechanisms that needs to be overcome there. I think a lot of research is being done on the adjuvants for those vaccines. Because if, let's say, vaccines have been shown to be able to induce the T-cell response and that you have them in circulation, but once they infiltrate the tumor, then if they're not efficacious, this is not sufficient. And that's where the vaccines might need a combination partner potentially in the future. So I think
Thanks a lot.
It's still a bit early in there, but that's definitely something that will be interesting to see progress in the next few years.
Thank you very much, Tarfa.
Thank you.
Ahdab.
Yes, can we take Dr. Salman next, maybe? Dr. Salman, do you hear us?
Yes, hello, can you hear me?
Yes, we can hear you, Salman.
Yes.
All right, so thank you for the interesting webinar. So I'm not an immunologist and nor a cancer biologist, so I'm more into diabetes and cell signaling. But nonetheless, it's a very interesting topic. So I have a question basically about solid tumors, so and hypoxia. So with immunotherapy, how effective is this approach towards these kind of cancers? And for the penetration, does it get affected with the level of oxygen? So if you have a solid tumor that is, let's say, quite huge in diameter, so if this kind of approach is affected or effective against it, do the T cells penetrate that far into these tumors or not? So that's one question. I do have a second question. Basically, it's about the way the immune system interacts with its target. So the cancer cell system is known to communicate via different means, so physical ways and also chemical ways. So for example, exosomes and peptides, and they change the levels of hormones. So can the immune system read these kind of signals, you know, on the fly and react to it or not? So these are my questions. Otherwise, thank you very much.
Thank you, Salman. Thank you very much.
Thank you. These are very good questions. I haven't touched much on these. Maybe I can start with the hypoxia. I think there it probably depends from patient to patient and the degree of hypoxia. There's a lot that has been studied on the impact of hypoxia and how it generates an immunosuppressive environment. So this definitely contributes to lowering the T-cell efficacy within the tumor. Approaches to overcome this, I don't have things coming to my mind right away. I believe there has to be multiple approaches also trying to tackle this, because that's also a major topic. But there I would not be the expert to provide you a complete answer.
So Salman was asking the second question about the means of communication in tumor cells compared to immune cells.
And in terms of communications, that's also extremely interesting and complex. You mentioned exosomes. I think there the research is also blooming. There's a lot that we need to still understand about this. The T cells can sense many things in their environment. Exosomes, I'm not aware of specific signals that have been shown going through that path for the T cells, but I wouldn't be surprised if this plays a role as well in either helping them or dampening their responses, depending where the source of the signal comes from. I think there's a lot of knowledge on chemokines and cytokines, maybe because it's a little bit more simpler to study in the lab, as we can synthesize them and test in various conditions. The exosome field is still puzzling me a lot, as we don't really know the content of those exosomes so much.
It wouldn't surprise me to say that from other systems, I mean, from the field I study in the nervous system and the immune system, there are common shared receptors where you'd see bilateral effects between two different systems. So if I'm understanding Dr. Salman clearly, he was saying whether hormones can affect the way immune cells act within the cancer. I know he wants to add one more point. Can you hear us, Salman?
Yes, I can hear you clearly. Yeah, this point just came to my mind. So it's about the microenvironment or the local effect of signals and the systemic effect. So I was just thinking, now we're approaching the month of Ramadan. So, you know, fasting basically has a lot of effects on the entire body, including inducing autophagy. So since we're in the context of cancer immunotherapy, does that actually aid and synergize with this kind of therapies? Does it help the immune system in doing its job? That's my addition. Thank you.
Thank you, Salman. Well, Ramadan is around the corner, Ali, so your advice will be taken.
I'm not going to provide any advice on it. I think that's definitely a personal choice that everybody should take. It's a very interesting question, and I haven't read literature on this. The impact of fasting on the immune system generally is something that is, I believe it has an impact on the immune system. The diet has such an impact on our bodies.
I don't know either, Ali. So
yeah, I think it's a very good question. At the same time, during Ramadan, it is not a complete fasting.
You don't eat for 9 hours.
It's only a timing, exactly. So your bodies still get nutrients that are needed to support the immune system. I do not know of the impact.
Okay, we'll let you off the hook for that.
There might be some good studies on it.
Yeah. Definitely push the audience to go and read about it.
Yeah.
All right, Ahdab. Anything from the chat for us?
Yes. I'm just looking at the chat.
Okay.
I do have also a question about, in general, what do you think about where we are now with cancer immunotherapy and the future of immunotherapy?
I'm very glad you asked this question. I have an interesting slide on this. It goes beyond cancer immunotherapy. But maybe to start with that, I think we're at the early phases of a revolution. What we've seen in the clinics with checkpoint blockades is only the beginning. On this slide here, what you see is the big trends of the evolution in pharma innovation. And if we look from early days, we had defined molecules, so mainly small molecules with a rather undefined molecular target with a phenotype that we knew was desirable. That was the first big wave where the research was maybe a little bit slower than now, since now we have amazing technologies to support research. And then that brought better defined molecules and molecular targets. So this is a one-to-one concept. So one drug, one target. And this is still something that is ongoing. And now we're entering the biologic fields since maybe yeah, let's say 3 decades or so. And there, the possibilities are constantly increasing. In terms of biologics, the combinations that can be done with various proteins, you can combine antibodies with cytokines to target them. You can combine antibodies with chemotherapies as well. So there's a lot of things being combined at the moment. And the new generation is then multi-specificity. So for example, the checkpoint blockade that are currently in the clinics are still part of the biologic wave, as they're specific, for example, to PD-1 or PD-L1. The multi-specificity is the next wave that currently we're seeing in research, and that's also part of what I'm doing, is, for example, taking a checkpoint that you combine with a second target, and then you have really this multi-specificity that brings more, that brings a biological impact localized to where you want this impact. So this is really something new.
Do you mean the bispecific antibody?
So bispecifics are part of it. That's one example, as one of the first that kind of opened that field. And in terms of specificity, it can be T-cell engagers, but it can also be dual co-stimulators, for example, and other multi-specificity drugs. So I think there, at the moment, it feels like that the sky is the limit, and it's still in early research, and then the clinical trials will be very enabling those drugs in the next few years.
And it's in line with all these new approaches with CRISPR as well, specified medicine and specified gene manipulation. Can I ask you, Ali, and then I'll allow one of my students to ask a question. Another student of mine, Saif Al-Harathi, was asking, what do we know about NK cell harnessing in fighting tumors? So, I mean, I've read some papers published last year. One I remember is by Minito and colleagues, where they say that NK cells are better for targeting the HLA-I negative tumor targets. Can you elaborate on that?
Yes, sure. I think that's a very good question. The NK cells are also part of the panoply of effectors that we have to fight cancer. Indeed, they are very efficient at killing cells that lose the HLA molecules. If we go back to the escape here, so the MHC or HLA loss is one of the major contributors to the escape. But NK cells so the NK cells can kill any cell that would lose the HLA very efficiently. So the NK cells really continuously monitor this. So that's a very interesting cell type as well to harness in cancer immunotherapy, as they're very good killers as well. And they're the way to do it, or the drugs to use this mechanism, are mainly focused, I think, at the moment in this multi-specificity field, since the NK need to be driven towards the target. So if, for example, you want to kill a tumor cell, but that still has the MHC, but has a loss in the downstream, so in the processing mechanism.
Intracellular cascades.
Exactly, in the digestion of the peptide and the presentation machinery. Well, then the NK will not kill the target cell because the MHC is still present on the surface.
So two different kind of mechanisms here, two different approaches for each cell.
Exactly, and the NK cells also have other mechanisms to recognize target cells. I think that's one of them is via the CD1D, which is part of or it has an MHC-like molecule, which presents other types of targets for NKs.
Mm.
So in the bispecific fields, then there are NK engagers, and there are also approaches in the CAR field, looking at making NK CAR.
And how far are we in that?
How far are we from the first clinical trials, or from saving the first patient?
First clinical trial. Yeah.
I think it's still a matter of years. I think but I don't know. I believe there's a lot of innovation that goes extremely fast in the biotech world, in the smaller biotechs. They can act very quickly on novel findings. I think maybe they are faster. I'm not aware of the current status on the trials for NK-related.
What you were saying, Ali, about which is one of the natural functions of NK cells, the killing cells that do not express MHC1. It's interesting how that can be harnessed then to fight cancer cells, because reducing or downregulating MHC1 is it's one of the viral mechanisms of evading detection, and NK cells are very good at fighting cells that are unable to express MHC1. So to see how that works in the tumor microenvironment will definitely be very, very interesting. Hadab.
Yes. I think I'll just add on the CAR, the NK cell, CAR NK cell. So do they think that the CAR NK cell is better than T cell in terms of the attenuated side effects, like to overcome the GVHD issue or cytokine storm that been associated with the T cell based treatment?
That's a very good point.
That's a very good question. I'm not aware of side-by-side comparisons of NK and T cell CARs on these aspects. T cells and NK cells are both very good at killing their target. They both have different co-stimulatory and co-inhibitory receptors, and they both can secrete quite some amount of cytokines that will be inflammatory as well.
Mm-hmm.
I do not know if there's a better prospect for one or the other cell type.
I think, Ali, the main distinguishing difference is the clinical efficacy of T cell based on immunotherapy. And that presents a series of limitations, including its inability to recognize and kill HLA-1 negative cells. I think that is the most distinguishing hallmark there between NK cells.
I mean, I agree with you, but let's say the scientific community and clinical development of molecules has shown very innovative ways to bypass that. The bispecific field being one of them, to then still have T cell killing, but without relying on the MHC, for example. I think these are very potent molecules that we didn't discuss deeply during the webinar so far. And that's actually one of the fields where I'm involved a little bit more. So these are then the T cell engagers. And I think these also have a very promising way ahead. And they can be for T cell engagement, but some can also generate NK cell engagement.
Adab.
Great. Thank you very much, Ali. We are really enjoying this discussion. I do have one more question.
One last question. Promise you, Ali.
I still have time.
Because we really enjoyed this discussion actually, and it's very stimulating. So because we are in the time of Corona, what do you think about the risk of SARS-CoV-2 infection in cancer patients?
That's a very good.
Do you know any like strategies that have been used with the cancer patients?
I think the pandemic has a lot of impacts worldwide on how we do research and early development, and also how the patients are being treated. For the cancer patients, I think it doesn't necessarily change their therapies, right? It's just that some of them have maybe a higher risk, so they might be part of a risk group to develop more severe effects when the therapies, for example, are immunosuppressive. Chemotherapy, for example, can have a very damaging impact on the immune system. And this could make a patient more vulnerable. So I think the approaches are really to protect the patients in the best way possible. I think so far the approaches that have shown the most success are isolation and social distancing. And I believe that that's likely to be what is also being done in the hospitals for those patients.
Great.
I think at the moment we're still eagerly awaiting the results of well-controlled clinical trials to see if we have efficacious drugs against that virus. Yeah.
Ali, I promised you one last question, but what I meant was this coming question would be the one last question. So it's one of my students, Al-Abbas, and he's been waiting for quite a while. Al-Abbas, what's your question?
Assalamu alaikum. Hello, everyone. Hello, Dr. Ali.
Hi.
It's been a nice discussion. I would like to thank you. And it's amazing and interesting.
Thank you.
I have a bit of a general question. What's your opinion in anti-neoplastons therapy, which are peptides that are found in the blood normally, but it's synthesized in vitro also in the labs? And perhaps you've heard about Dr. Brzezinski in the state of Texas. He ran a clinical trial since 1970. And I heard that he had cured 2,100 people. Some of them were at the fourth stage of the cancer. And some of them were hard to cure. What's your opinion about this topic? And do you think that in the future drug companies would focus this therapy, studying more and doing more research?
Thanks a lot for your question.
Thank you, Abbas. Thank you.
Welcome.
I'm not sure I understood it correctly. So you said peptides that are in the blood. So these are synthesized peptides that are anti-neoplastic. Is it?
Yeah. For example, like phenylacetylglutamine and phenylacetate, phenylacetylizoglutamine. They say that it induces the immune response. It's a peptide that induces the immune response.
So Abbas, what you're saying is you introduce these peptides to induce an immune response against the tumor?
Yeah.
As part of a vaccine, or how do you introduce them into the patient?
They are synthesized in a lab. We know a little bit about it. I couldn't find information about it. There's a doctor called Stanislaw Brzezinski. He's a Polish doctor. He discovered it at 1970. I think there's a way that he combines these peptides to make a drug, a certain drug.
As part of conventional medicine, conventional therapy. Okay,
Yeah.
so I think what he's alluding to here, Ali, and you can correct me if you understood differently, is combining immunotherapy or conventional cancer therapy with immune adjuvant peptides. So something we see in other components of treatments as well. Is that being considered, or is that something you know of? Thank you, Abbas. Thank you very much.
Welcome.
Yeah, thanks again for the question. I'm not actually aware of the research that is being done on such peptides. I know that then in terms of adjuvants, there's definitely a need for something that is safe and supporting good immune responses against cancer. Which are then combination partners in multiple approaches.
Do we need it in our approaches? Just a side question. Do we need adjuvants in immunotherapies?
I think it really depends on the combination partners that one would consider. We were talking about vaccines earlier. Maybe an adjuvant, a better adjuvant could increase the vaccine successes. In terms of, if we look at T-cell engagers, combining with adjuvants might not be the first thought, but still might support building an immune response. So the adjuvants usually can be seen as the danger signals that are then taken up by the DCs, so the antigen presenting cells, that can in turn activate the T cells. So if the DC is sensing a lot of danger signals, then there's a better chance that the T cell will be primed against an antigen. I think those peptides might have a chance in that direction, but I'm not aware of the progress on that front.
Okay, well, Ali, thank you very much for the time you gave us today. I would like to first thank my co-host, Ahdab Al-Saidiyah. Ahdab, are you there?
Well, thank you very much. I don't have anything to add. I just would like to thank Ali for this great talk and stimulating discussion. Thank you for your time.
Thank you, Ali.
Thank you very much for inviting me. I'm very glad to spend some time with you all and to be able to share some of the excitement on the new research.
Yes. Well, Ali, you've been more than generous. I've known you for a nice decade now. You've never looked younger, while I've aged horribly. I want to thank you again, and I would like to say that I appreciate the time you've given me and my students and all my colleagues here to learn more about the new and emerging field of immunotherapy. Anything you want to say at the end?
Just a thank you for everyone for all your questions, your interest in the field. Stay tuned, and there will be more progress in the next few years.
Thank you very much. Okay, thank you, Ali. Thank you very much, everybody. See you all very, very soon. Bye-bye.